Hi girls, I'm going to find you some bonus questions. Yay, we've got some bonus questions to do with signalling molecules. So the first one, we're going to get started. This is a bit of an old study design question, but it's still, we can work through it and get it right. So this is from 2015, the exam back in 2015. And we're going to say, which one of the following is true for chemical signaling molecules? And I want you to say, do they always produce a rapid response, produce a response in target cells when released, only stimulate an effect on the cell that produced them, or do they require carrier proteins to be transported through bodily fluids or body fluids? So I want you to pause and then come back and answer. All right, so now you're back and we think about, do they always produce a rapid response? No, these guys are chemicals. Chemicals take a long time to diffuse, so they are slow. So that one's going to be wrong. Do they produce a response in target cells when released? Yeah, that sounds pretty good, yeah? So we'll go on to the next one. Do they stimulate an effect on the cell that produced them? Sometimes they can do that, but not all signaling molecules can do that. Only the autocrine ones can. And do they require carrier proteins to be transported through bodily fluids? No, because carrier proteins, sorry guys, carrier proteins are embedded in the plasma membrane. They've got nothing to do with your fluids or your interstitial fluid. So therefore that one cannot be right. So therefore our correct answer is B. So this picture here shows cells involved in detecting and responding to itch stimuli. Itchy, itchy, itchy. Now that I'm saying the word itch, I'm sure you're all itching. So an example of the itch stimulus is the bite of an insect, which can result in itchy skin. Now a response to itch is, of course, to scratch. So using an arrow, show the position of a synapse. Now you used to need to know a lot more about neurons, but I think we can still get there. So a synapse could be here, a synapse could be here, or a synapse could be here. Any one of those three, I'm perfectly happy with. Now, describe how a signal is transferred across a synapse. So I want you to pause and I want you to think about your answer, and I'm going to show you. So here we have our positions of our potential synapses. We can see here that we get an average of 0.9. It was a pretty easy question. But then this one got a little bit trickier. Now, we used to need to know about calcium ions entering the axon terminal. But more importantly, what I would want to focus on when we're talking about how a signal is transferred across the synapse is I would say that we would talk about the neurotransmitter. This is the new study design format. Yeah, the neurotransmitter was going to be released. Then we're going to get diffusion. We're going to get binding to the next receptor. We're going to get, and that's an extracellular receptor. Now let's go back and have a look at this answer. So the neurotransmitters are released, they diffuse, they attach to receptors on the postsynaptic, which means on the other side of the synapse, that side of the membrane. So I think with this, what's important is, even though this is an old study design question and you don't need to know about calcium ions anymore, we could still get the marks. We could still get 3 marks based on our knowledge of the way that neurotransmitters diffuse across the synapse. All right, this is a pretty typical question when it comes to hydrophilic and hydrophobic molecules. So I'll get you to pause and then we'll go through it. All right, welcome back. So scientists are currently studying the molecular mechanisms that cause itchy skin. It has been discovered that many different receptors and signaling molecules are involved. So we've got different receptors, different signaling molecules. 2 specific itch receptors, H1 and TLR7, have been identified and their cellular locations are shown in the table below. Complete the table by suggesting the chemical nature of the signaling molecule and justify your response. So the receptor for H1 is on the plasma membrane. So what is the chemical nature of that one? It's going to be hydrophilic. Whereas the receptor for TLR7 7 is going to be inside the cell, so it's going to be hydrophobic. And then we need to justify. Now, I can't type that fast, so I'm just going to show you the answers here. So you could say hydrophilic, you could have said polar, you could have said protein. With the other one, you could have said lipid, you could have said hydrophobic, you could have said non-polar. So all of these words, you don't need every single one, you just need one, but they are all accepted there. You can see 36% of people got nothing, and 47% of people got everything. And then in the middle, it really didn't happen. So it's a pretty poorly written question to end up like that, but that doesn't matter. These are the types of questions that we've seen year after year. So why did we think this first one here was protein-based? Because it was on the outside of the plasma membrane. Yep, which indicates that you can't get through it. Why did we think this one was hydrophobic? Because it's in the cytosol, which means that it can cross or diffuse past the tails or something along those lines. All right, another question. So have a pause, have a read, and then come back. Okay, so after exercise, low levels of blood glucose can result in the release of glucagon, which is a hydrophilic hormone. Okay, if it's hydrophilic, what do we automatically think? Extracellular receptor. All right, so this is from the pancreas. One site of glucagon activity is liver cells, where it causes a process called glycogenolysis to occur. In this process, stored glycogen is broken down and glucose is released into the bloodstream. Describe how hydrophilic hormones, such as glucagon, stimulate a metabolic process, such as glycogenolysis, within a cell. So this question, it doesn't matter about the glycogenolysis. It doesn't matter about glucagon. It doesn't matter about all of these different words that we're going through. All that matters here is that you can talk about how do hydrophilic hormones stimulate a response. So what do we want to go for? You need to say it binds to the receptor on the cell membrane or on the outside of the cell membrane. The signal is transduced by a series of secondary messengers. Then after that, you end up with a cellular response. And that's it. Now, I know they haven't written this in dot points, but you could so easily write that response in dot points. Yeah? Don't make life hard for yourself. Continuing on with the question, when glycogenolysis occurs in a cell, the concentration of glucose in the cytosol increases. So here we have, automatically, I think that's going to be a response. Okay? Glucose passes through the plasma membrane by facilitated diffusion. Old study design, but we did it anyway, so let's try. So explain how facilitated diffusion of glucose occurs. I'm going to tell you right now, you need about 5 words to answer this question. So give it a pause and try. All right, so hopefully you're back and we can think about what are our 5 words. So the first ones we're going to need are whether or not we have a protein channel. Good, that's 2 words. The next one is high to low. I'm going to need 7 words, 8 words, sorry. Conc of glucose. They gave you all those lines, and these are the 2 points that you need to say. Let's finish off on this last question. Which organelle would you expect to see in large numbers? Sorry, muscle cells. I didn't do the reading of the initial bit there. Muscle cells in the heart contract and relax more rapidly during exercise and require a constant supply of energy. Which organelle would you expect to see in large numbers in heart muscle cells to supply this energy? Okay, this isn't about cellular response stuff, but we got to link together our concepts. Explain your response. So pause and tell me which organelle. I'm really hoping that you're unpausing now and you're coming back and you're going to say that you would have lots and lots of mitochondria. Why would you have mitochondria? Because they are the site of aerobic cellular respiration. Lots and lots of ATP for moving. It's not good enough to make any of those connections without finishing it off. Okay, so it's not good enough to just say mitochondria because they make ATP. No. Mitochondria, the site of aerobic respiration, because these make lots of ATP, and even that's not good enough. You need to link the ATP to the fact that they can do lots of muscle contraction. Okay, had to mention the ATP. And that is the end of my little bonus question round.